Hi, we're just going to wait a few minutes for everybody to join. All right, great. I think we'll get started. So I'm Merit Cudkowicz. We're here. We have a lot to talk about today, so we might go past our 5:30. So hopefully that's okay for most people. Before I start, I just want to welcome a few guests, special guests here. Vulcan Granite and Kathy Grossman from Biohaven are here, and Melanie Quintana, who's our lead statistician for the HEALEY Platform Trial, is also here, and Lindsay Pothier, who is a senior project manager for the HEALEY Platform Trial. And I think that people are very familiar with Jinsy Andrews and Suma Babu, who are the co-leads for the Biohaven trial. And then Allison and Catherine are here as they are every week. So first, just a pause to hopefully everyone has seen the wonderful news that the FDA has approved AMX 35. So we'll just take a moment to yay, it's huge. And this really was a community effort to have that happen. So great news. And I'm sure there'll be many more webinars and talks about that and what the next steps are for that. But it's a great moment. With that, maybe we can pull up the slides. And we wanted to show the high level results of the Verdiperstat, what we call the Regimen B trial in the platform trial. There'll be other opportunities to go into more depth, but since we announced the results this morning, we wanted to use the opportunity of this meeting to share some of the results. So next slide, please. First of all, many of you are part of the platform trial as well as other trials, and I want to take this chance again just to thank you for being part of research and being part of trials, because we can't get to approvals or can't get to new treatments and new understanding of the illness without your being part of it with us. So thank you for that. Next slide. As you know, the platform trial is an approach to try to speed up getting answers about whether a drug works or not. We're currently testing 5 different treatments, and we share the infrastructure and we share the placebo data between these groups as a way to get answers faster and with fewer participants. And just a moment to really thank Dr. Andrew, who's the lead investigator for Verdieperstat, and Dr. Suma Babu, who's the co-lead. Maybe I'll give them a chance if they want to just say a few words, maybe Jinsy.
Yeah, thank you so much, Merit. It's been a pleasure to work with the entire team, and it's a massive effort, truly a community effort to get through the trial and get to our answer efficiently, and really grateful for everyone's participation in the trial and communicating together to get this done. Suma, do you have other comments?
Thanks, Jinsy. I share Jinsy and Merit's sentiment about everybody's participation. I'm very grateful. We're all very grateful for everybody's participation. It takes more than a village to run these trials and get to the answers that we're looking for to help the entire ALS community. So thank you so much for participating.
Thank you, Jinsy and Suma. Of our centers, 54 of them were participating in Regimen B, and they're shown here. We did meet with them this morning, the site investigators, so that they could see the data and call the participants and share the results and kind of next steps as well. Next slide, please. So Verdiperstat is a myeloperoxidase inhibitor that targets microglial activation. We started the dose at 300 milligrams and then went up to the target dose of 600 milligrams twice daily by mouth, and that was after a 2-week titration. Next slide, please. I'll just go through what the primary measures were, but then ask Dr. Quintana if she can walk us through some of the results. So the primary outcome measure was disease progression as measured by ALS Functional Rating Scale through 24 weeks. Secondary measures was also looking at the ALSFRS using a repeated measures model, looking at strength using something called handheld dynamometry, both in the upper and the lower extremities, and looking at breathing function. And so today we have what we call the top line results. But we still have other analyses to do. These are called exploratory, and they include things like the digital apps that people were using, the voice measures, and the home breathing, fluid biomarkers, and some other clinical assessments as well. And once those are all available, we will be sharing those broadly as well, and obviously writing a paper on it. So I don't know, Melanie, if you want to take from here. You're muted though.
Sorry about that. Sure, I can start from here. Just showing here in the diagram, how many individuals were assessed for eligibility and how many were assessed for eligibility specifically for Regimen B. Just want to make a note that we're going to be showing, I believe, results from the two main primary populations. One we call the FAS. This is the regimen treated and the regimen specific controls, as well as the shared controls from the other regimens. We'll also show results for what we call the ERO. That's the regimen treated and the regimen controls only. So you'll see those at the top here, the FAS, and then you'll see the ERO as well. 167 were randomized, 3 were excluded due to screen failure or early termination. 122 total randomized to placebo across Regimen B and the shared placebo, and then 126 randomized to the active treatment. Next slide. This is the main figure really of the results. The primary endpoint for the platform trial is change in disease progression over 24 weeks as measured by ALS-FRS-R. The primary analysis, as recommended by the FDA, accounts for mortality. So it's a Bayesian shared parameter analysis of function and mortality. Because we expect mortality to be low, the major component to what estimates a treatment effect is the ALS-FRS-R change. So that's what we're showing here. In the blue circles and the blue line, that's estimated for the treatment group. The gray squares are the raw estimates for the Regimen B only placebos. The black triangles are the pooled placebos across all regimens. And the gray line is an estimate of Regimen B placebo with sharing from the black. So the primary analysis model shares from all the controls and gets an estimate for this regimen using all the information. And the main thing to see here is that really the progression for the treated and Regimen B with sharing from the other placebos is right on top of each other. So very similar progression rates in those two arms. I'll also just note quickly that the model adjusts for potential differences in baseline, such as time since onset, pre-baseline slope, edaravone use, and riluzole use. Next slide. Merritt, do you want to go back to this slide? Okay, thank you.
Yeah. Thank you. Yeah, so the main outcome measure you just saw, and that was negative here. We also looked at all the secondaries and did not see any difference between the treated group and placebo. And so those included measures of muscle strength, respiratory function, as well as survival. As I mentioned, there's still a lot more analyses to do, including the fluid biomarker subgroup analyses and the home assessments as well. And we're hoping that those will be completed in the coming months and presented at an upcoming scientific meeting. I did see in the Q&A that there was a question about which data sets were used for this. So each of the regimen partners were given an option about whether they wanted to use the shared placebo group from all 4 of the regimens who were starting out, A, B, C, and D, or just A, B, and C. And for Biohaven, the decision was just to use the data from regimens A, B, and C. Next slide, please. So we did want to share, and this again was shared with the sites and we shared with the participants, that because of these results, we are going to stop the open label extension and the expanded access protocols. We didn't show you here all the data we have on safety, but we did not find any new safety signals. So we think that this can be done, given people's schedules, hopefully in the next 6 weeks. The sites should be calling the participants, and we will be having a participant webinar next week to, again, thank the people who are in the Regimen B and show them more of the data. So we did release this information this morning. We are sharing it here publicly for the first time. We shared it earlier with the site investigators and people on the steering committee and some of our vendor partners so that they would all know. And we're going to have a participant-only webinar next week. We're starting a writing group so we can get the data out. No matter what, we've designed these studies to keep learning about the illness. I think that's really important. Of course, we're hoping for a positive result, but it wasn't positive, but it's going to keep informing about the illness. And maybe before we go to some of the questions or some of the updates on the rest of the platform trial, I wanted to give Kathy and Vulcan a chance if they wanted to say anything. They're from Biohaven.
Sure. Kathy, may I go forward first?
Absolutely.
As good news and bad news happen, as we all see today. Amylyx drug was approved. We're very disappointed that we were unable to add yet another option for our patients. And some of you in the audience may remember that I'm also a site investigator and an ALS doctor who is now working for Biohaven. So I feel the disappointment really deep inside that this didn't work. And we're extremely appreciative of our patients for being in the study. So we learn by, both winning and losing. We understand the disease by learning in these clinical trials. So thank you very much. And it's been a great experience working with the HEALEY Center. So we also appreciate that opportunity, and we wanted to publicly thank all of you.
Thank you, Vulcan. Kathy?
Yeah, thank you, Vulcan. I don't have much more to say except for that it has been an honor for Biohaven to participate in this very important trial. Our commitment to learning and bringing forward new treatments for these rare neurological diseases will ongo. We have faith in the platform trial strategy and design that hopefully more treatments will be studied and come out with options at a faster rate. And we have so much gratitude for patients and their caregivers and family members. Without your support, your time, and efforts, none of this could be done. And we know the desperation that's needed in this patient population, and we appreciate all of you.
Thank you, Kathy. And now, Sabrina, before the questions, do you have any updates on the platform trial in general, or would you prefer to go straight into it?
I think we can go to the questions. Yeah.
Great. Maybe do you want to be the pilot?
Absolutely. First of all, thank you for all the messages. There's a number of messages about Amylyx and very nice messages. So thank you so much. A few questions now, staying, keeping the topic on the platform trial and the Verdieperstat regimen. Were there? One question is: were the outcome measures for Verdieperstat for phase 2 more positive than I guess for the current trial? I think that's the question. Were there previous data, I guess, for Verdieperstat that were more positive than what we saw today?
I can answer a little bit, but then obviously Vulcan or Kathy. I mean, the reason why our therapy evaluation committee was excited about bringing this drug forward is that the target of the drug is one that we think is very important in ALS, and that's the microglial activation. And Biohaven had data using an imaging approach, a PET ligand, to show that their drug could get into the brain and hit that target. So I think the science and rationale were strong, but this was the first study, as far as I know, of their drug in ALS. They had a study in other neurological illnesses, but the first one in ALS.
next question. Have you, Katie,
Oh, wait,
Kathy,
did
do
Kathy
you want to
want-
add anything?
No, I was just going to say we agree. And yeah, hopefully, the biomarker data that is forthcoming may help us. But unfortunately, clinically, it didn't show positive change.
The next question about this regimen, have you analyzed the data from the open label extension?
And no, we have not yet. No, and that's we would have to do the open label analysis. We would have to also lock that database, and the decision to stop OLE it couldn't be made until we saw the results. So now that we know that we're going to stop the open label extension, once the last person has come for their last visit, we will be working on locking that database and then doing all those analytics as well.
Next question. So now we have negative results for regimens A and B. So what have we learned to assist development of new treatments from this trial so far?
Yeah, that's a great question. I think, as we know, it's an illness where we need to learn more about the science so that we can keep getting better targets. I think we thought the science was good for both those drugs. And again, the platform trial was an efficient way to know that they didn't work. I mean, again, our goal is to find drugs that work, but we also want to find quickly the ones that don't work. And I think in that way, we learned that. I think, for the first one, I think it does tell us that for regimen A that hitting complement was probably, that part of the complement system is probably not effective for ALS. And I think here, we're also learning that myeloperoxidase isn't a good target. But we need to finish some of that biomarker work to really know that we hit the target in the way we wanted to, and we've ruled out that pathway. The Amylyx, I would just say the Amylyx positive was after a lot of drugs that didn't work. And so that is a huge finding, but we still need to invest more in the science and understand the biology so we can keep getting better and better targets and drugs.
So there's a number of questions about timelines, and so I'll summarize them because they're fairly similar. So you mentioned that regimen B used shared placebo for regimens A and C. And so there's a number of questions about when we will hear about C, how about regimen D, has this been completed or not?
Yeah. So I know every week we ask these questions and I hedge a little bit. I just want to say the reason I hedge is that there's actually, federal laws that prohibit my telling you these kind of things. So when, as you saw from today, we announced the results in that press release, and that's required by SEC rules that we have to do that. And then once that was out, we really tried simultaneously to let the sites know, let the foundations know, let participants know, but we can't do it in advance. So I cannot tell you on this call when we will be sharing the results of C or D. I can tell you though that we're working all the time to do it as fast as possible.
There's a question about futility analysis and our interim analysis. So as you know, that we have interim analysis for all the regimens for futility. So the question is, was this analysis done for regimen B, and did that support continuing the trial regimen B to its completion?
Yeah, so the futility analyses are done on all the regimens, and again, I'm a big proponent that every ALS trial should have that. But we do have one of the experts in this, the statistics behind it, and maybe Melanie, you could help answer the question.
Yeah. So the futility rules were designed through clinical trial simulation, and our main goal is that we would never want to stop a trial that could go on to be positive. So when we looked at our simulations, we created a rule that was our main goal. We never wanted to stop a trial for futility that could go on to be positive. And because of that, the futility rule, I would say, is rather conservative. You could call it conservative, and that it really would only stop at one of the interims if you were starting to see a trend in the negative direction. And so because we obviously never saw that here, and we didn't even see that at the end, that's why this regimen did not stop for futility. And I honestly think that's for good reason, because we really want to give it the benefit of the doubt and make sure we watch for as long as possible to see what could happen.
I'll just add to that this is a short trial, so the 6 months, and we feel very strongly that most drugs 6 months is the right amount of time, because we are asking people to be on placebo. But if you abort with not a lot of follow-up data based on a futility, you could be throwing out a drug that might be effective. So that's was some of the reasoning behind being conservative.
Actually, there's a question about trial duration, and the question is, what if future data, like open label extension data or additional biomarker data, seems to be more positive than the top line results that were shared today? What would happen then?
Yeah, I might ask, I don't know if Falcon or Kathy want to weigh in on that question.
I apologize, I couldn't hear the question.
Oh, can you repeat that, Sabrina?
Yes, absolutely. So the top line results that were shared today are about the placebo-controlled portion of the trial, which was 6 months long. And then obviously we're going to analyze more biomarker data as well as longer term open label extension data. So what if those data are actually positive or better than what we shared today? What would happen?
So I want to start that is an unlikely scenario based on the results so far, but we may be proven wrong with the biomarker data. However, this was repeatedly discussed in our internal calls that we are committed to providing this to patients in the appropriate settings. And if the results are positive, we will continue to further investigate and understand why we didn't see what we wanted to see clinically, and if those biomarkers results have a clinical relevance, and if so, we will continue to investigate.
And Biohaven's goal is to be able to provide what we can that makes sense and is safe. So if data comes back saying something other than what we're thinking right now, which there's a lot of analyses still to do, we will definitely take measures to get it in a responsible way to people who could benefit from it.
Great. Actually, a question about, there's a separate question about options for trial participants, and specifically with respect to the people who participated in Regimen B. Obviously, they dedicated time, energy to participate. What would be, what are the options for them? Can we facilitate access to other investigational products for them?
Well, so anyone in the trial that is, let's see, still eligible for the trial, still meets the criteria, could go into another arm if they wanted to. They would have to be off of Verdieperstat for 30 days. There's also lots of other trials. I mean, an enormous number of other trials that are going on right now throughout the United States and the world, really. So it would be about maybe talking to your physician and your site about whether there's something that you're eligible for. Obviously, the Act for ALS initiative is about funding expanded access programs, and we should be hearing soon about that. So there might be some expanded access programs. As we get results for C and D, I think depending on those results, if anything's positive, maybe those companies will be open to an expanded access. It's hard to predict that yet without having those results.
Question about biomarkers. Was PET used as part of this regimen?
We didn't use it in this study because it was done actually before. So it's better to do PET in a small study first and see if you get the right dose and that target. And so that was done already. So there was no need to do it again in this study.
Great. So there's a question about statistics. Regarding the placebo, can industry partners choose whether they want to use the pooled placebo or just the regimen-specific placebo for the primary analysis? Maybe I don't know, Merit or Melanie, you can explain a little bit in terms of the choices about primary, secondary, and use of placebo data.
Maybe Melanie, you can answer that first.
Yeah, sure. We recommend for the primary for all regimens to use the shared controls. The major question is whether you want to use shared controls from a regimen who is not going to be completed at the same time as yours, so at a later date. And so when Merit brought that up earlier, she was speaking with regard to B choosing to use D, because D, they would have to wait some time then for D to be locked. So that's a reason they got to choose whether to use D or not. But the primary does recommend using all shared controls, with a question of using controls from still what we call open regimens or not.
Great. There's an operational question about timelines. So as you know, we have these webinars every week. And so in August, we said that some of the databases were locked. So the question is, what is the gating factor to finish the analysis and release the results?
I think the gating factor is being done with them. I'll just give an example. I think there were over, I don't know, 3,000, maybe 4,000 tables involved in this regimen, and that's going to be the case. It's a lot of data, and it takes time to make sure it's done and it's done correctly. And so every study is going to be a little different, but what you want to do is make sure that you're confident in the results before you share them.
Great. So there's a question about combination of different drugs. So Amylyx that was approved, AMX35 that was approved today is a combination of 2 drugs. And now we have in the platform trial, the negative regimens A and B, we're testing 1 single drug in each. So can you share your thoughts about combination of drugs to exploit synergism?
I'm happy to share mine, but I don't know if Suma or Jinsi want to add too. I'll say that I think that for sporadic ALS, that until we know more about the most upstream cause, we're going to need combination of treatments. And in a way, these trials were all combinations of treatments because people could be on riluzole and Edaravone plus a new drug. And future trials will be riluzole, Edaravone, and Amylyx drug. So we are doing combinations. The Amylyx thing made sense because they did in the preclinical space, like before people, they did studies that showed their 2 drugs were synergistic, that they were better together than either alone. And I think we'll see more of those types of drugs, but you have to do those studies because sometimes drugs can actually go the other way, that they can negate each other's effects. So it's really important to look at the whole picture that way.
Yeah, there's a question directed to you, Dr. Granit, specifically. So the question for you is, as a clinician from Miami, can you speak to the percentage of people who are Hispanic or African-American at the Florida sites? And maybe Merit, you and I can speak about sort of enrollment of diverse populations in the trial.
Yeah, so the University of Miami Clinic has a very large population of Hispanic and to a lesser extent African-American patients of ours. And I know for a fact in our studies, we have many patients who are Hispanic, including, in HEALEY. So, I think we highly encourage, like all of us, participation in clinical trials and observational trials so that we understand this disease, so that we find the most upstream pathology, as Dr. Cudkowicz said, to get the right target, and that's critical. So definitely, all are welcome, and huge Hispanic population in Miami.
Yeah, and maybe I can just add that definitely we have a commitment to ensuring a more diverse population than that's been the case in historical ALS trials. It's clearly there are many barriers, and we're trying to tackle several of them, including having sites that are geographically dispersed, so that obviously we can get enroll closer to where people are and enroll different populations. As well, we have a number of other efforts in terms of outreach. I know we have Catherine and Alison on the line, our patient navigators, and with them and others also on the line. And thanks to funding from the Tambourine Foundation and other groups, we're trying to again develop methods to be more equitable and again reach different populations. So certainly a lot of work that still needs to be done with that. Now, I know that we have one more statistical question. I know we always get statistical questions on this webinar. Many people follow this very closely, so I'm so glad you're on the line, Melanie. So the specific question is the pooled placebo in the HEALEY data consistent with the PROACT data of about 1 point decline per month?
It's nearly identical. It's nearly identical to that estimate. So yes. And consistent, you saw consistent the shared control with the regimen only control as well.
There's a great question about staying on medication or not now that this study is completing. For the people who are in the open label extension, the question is, would you want people to stay on the drug to measure biomarkers or not?
Yeah, it's a very good question. I think one is we do have, and again, thank you to the participants, we do have a lot of biomarker data, both in the double blind as well as in the open label extension that will be analyzed. I think for the people who are in the open label now, it's really a choice of whether to stay on the drug until your last visit, which we've asked the sites to do within 6 weeks so that we can get biomarker data while you're on drug, or if somebody wants to stop it before they come back for the visit, that's okay too. We do have a lot of data. More is always better, but we wanted to give people that choice, and that's something the site physicians and nurses that you know well will discuss with each person one-on-one.
Yes, there's a couple of questions about access and options. Maybe I'll ask Dr. Babu, since you are also involved in enrollment in many trials. So what are options for people who are looking for trials or perhaps expanded access? Who should they contact to see what's available?
Thank you, Sabrina. I think your first point of contact is to talk to your neurologist, to look at what options are available closest to where you live. And obviously beyond your local site, your local clinic, you can always reach out to contacts at the NEALS Consortium as well as at the HEALEY Center. We're here to help and support you through our patient navigators. And you can, there are also a list of trials that are available, and this is constantly changing, both on the HEALEY Center website as well as on the NEALS Consortium website. And clinicaltrials.gov is another great place as well to look for trials.
Great. Question, do you know if non-platform trials are committed to publishing even if they get negative results?
I hope so. They have to put the results up on clinicaltrial.gov. That's a requirement, and there's quite a hefty financial fine if you don't. So that for sure will happen. But I think whenever there's investigators like people you see here and our other colleagues that are part of those trials, that's really actually part of the contract, and we really push. So it's always published. We need that. And the other thing we try to encourage, a lot of companies are doing, is sharing the data with PROACT, so we can keep building that natural history or natural trial history data set with concurrent data. Yeah.
And going back to the question of access, maybe Dr. Babu, a specific question about expanded access. People are asking, if people are no longer eligible to re-randomized into another regimen, can you share some thoughts about EAPs?
The entire EAP space is growing, and it's rapidly growing, and we hope that it expands multifold from where we are at the moment. And currently, as much as we want every ALS patient to get access to an expanded access if they don't qualify for trials, I think the number of spots, the number of sites that are offering these EAP slots are quite small. And we hope that with the Act for ALS, this landscape will change, and a lot more ALS patients who are interested in participating in research will have access to the expanded access programs.
Yeah, I want to take this moment maybe to point out a great new initiative called Tech versus ALS that a person with ALS has started to build new EAP sites. So we just had a ribbon cutting at Jefferson in Philadelphia to be a new EAP site, and to have funding and support, and so they can have staff to do that and to enroll people in EAPs related to the platform trial, but also other EAPs. So hope is to do many more of those.
Yep. And people are asking, will there be a Regimen E expanded access? So certainly we're working on that. And are there provisions for any ALS trials to end early if the results are exceptionally good?
Very good question. I might ask Melanie to address that one as well.
We haven't added that into the platform just yet. We really wanted to learn as much as we could with these ones. So we wanted to follow everyone out the full 24 weeks, but we're definitely open to that in the future.
Great. I think we I've taken most of the questions I've seen. There's one question I would like to answer. How do we pronounce the name of Amylyx's new drug? And I want to say for the first time, Relivrio, I believe. So
Say it again?
I believe it's Relivrio. I mean, it's wonderful name, I think.
Sabrina, can you say that 10 times fast?
There's a few R's and I rolled the R being Italian, but anyway.
It is, it's an amazing day for that.
Yeah.
I'm sorry that we have this sad news as well, but we are determined to keep going. But it's a huge day in many ways. You know, another drug for people living with this illness, but also a community that came together and was heard by the FDA. And I think it's good. It's just phenomenal news.
Perhaps we'll end on that note, and certainly we'll be here again next week, like every week, and we look forward to seeing you next. Thank you to everyone who participated and to all the speakers.
Thank you.
Thank you.